So I hope you all had a fantastic snow weekend. Because snow like this only happens for Santa like once in 10 years. So my family really enjoyed it. I hope you get the most out of it. Okay. So it's really a great honor to have Dr. Xinzhong here with us today. Dr. Dang earned his bachelor degree in chemistry from Holy Cross College and went on to complete his PhD with Dr. Larry Ziprsky at UCLA. So Dr. Dang he's a postdoc training with Dr. David Anderson at Caltech. Dr. Dang currently is now a professor in neuroscience, neurosurgery, and dermatology at Johns Hopkins University. So he received the HHMI Young Investigator Award 2009 and became an HHMI investigator in 2015. Biology, his research has identified a family of G protein-coupled receptors that regulate pain and itch sensation, which has led to the development of novel anti-itch compounds currently in clinical trials. Dr. Dang is also a researcher on that one. Almost every postdoc in his lab has gone on to become an independent, and I was lucky to be one of them. So I'm looking forward to hear about his recent research about MRG independence.
Thank you very much for the nice introduction, and thank you for coming to my talk. And it's always the best feeling to see the former trainees achieve great success like Leon. And so Happy New Year, everyone. And so today I'm going to talk about this GPCR family. I cloned when I was a postdoc in David Anderson lab. And sorry, it's not down. Freeze here. Yeah, I think he put in there. Problem here is not moving.
This one's not moving.
Yeah. Okay, maybe I just do manually then. Okay, now it works. Yeah. Okay, great. So, yeah, sorry for the glitches. And so the MRG encode a large family of GPCR, and there are 50 mouse MRGs shown here in family and 18 human MRGs. And the interesting thing about this family is it only evolved very recently. It's only evolved in the tetrapod, the animal with the 4 limbs. Now it makes sense because only the animal with 4 limbs can scratch their itch. And so they don't exist in my fish, actually. So here, most of the MRGs are clustered on the mouse chromosome 7, the human chromosome 11. I show the gene cluster here, and each bar represents 1 MRGs. And the MRG was first identified in sensory neuron, and later on were found in other immune cells like a mast cell, neutrophil, and basophil. And based on the sequence, based on the expression pattern and also the ligand specificity, we can assign different human homolog shown here. So this gene family, as shown here, expressing diverse cell types and have a different biological functions. So let me tell you the story on the this receptor as an itch receptor first. So what is itch? Itch is defined by this German physician more than 360 years ago as an unpleasant sensation to make you want to scratch. And although it's a very simple definition, but it's still accurate, so we currently still use it. But why do we need itch? So itch is considered as a protective mechanism, just like a pain. And you want to scratch to remove irritant from the skin. And other people think scratching can actually mobilize the immune cells in the skin, so you have a more immune response. And also, itch can call our attention to the affected skin areas. and avoid the irritant in the future. And also the scratching can really provide temporary relief, so you feel good, so that's why you're scratching it. However, the pathological or chronic itch can lead distress and suffering, doesn't have much beneficial value for chronic itch. So the work from my lab and many other labs now show the itch is a distinct sensation from pain sensation. And also the beautiful work from Liang's while she was a postdoc in my lab and showed there's an itch-specific neuron in dorsal root ganglions. And we also find that this MRGs, the receptor we clone, some of them actually express in dorsal root ganglion sensory neuron, and these are the neurons send the axon to the skin. And some of these MRGs function as a novel itch receptor mediating non-histaminergic itch. So just give you an example. So this receptor is called the MRGD. It's a receptor for beta-alanine. So if you do bodybuilding, you probably heard of a beta-alanine because the bodybuilder tend to take a lot of beta-alanine and other supplements to build muscles. And however, they all experience very strong itch as a side effect. It turned out this beta-alanine can bind to MRGD to generate this irritating itch sensation. And besides those acute itch, when the itch lasts longer than like 4 or 5 weeks, that becomes chronic, and that's a disease state. And as you know, many skin diseases like eczema and psoriasis or different type of dermatitis can also cause a chronic itch. And then the systemic disease like liver disease or kidney disease can also accumulate a lot of metabolite in the system to cause a chronic itch. And drugs induce side effects. Itch side effects is also a big problem, like morphine, analgesic morphine can cause a lot of itch. Also, there's a neurological, neuropathic itch like shingles. And so over 2,000 years ago, the Greek physician made a striking notice, and they found a lot of yellow skin people tend to complain intensive itch. Now we know this condition is called the jaundice. So the jaundice is actually caused by accumulation of the yellow metabolite called the bilirubin and in the system. And the bilirubin is a degradation product from heme. And normally it can excrete it in the urine and the feces. And however, if you have a liver damage and this secretion pathway is blocked, the bilirubin can accumulate a very high level to cause a yellow skin and also cause itch sensation. And so basically the jaundice associated itch commonly occurred in the context of liver damage like cholestasis, like the blockage and stoppage of the bile flows. And so this cholestasis can cause by many different liver injuries. And many people in the US actually suffer from this condition. However, currently there's no effective treatment for cholestatic itch. So when a talented former MD PhD student, Jimmy, entered the lab, he wanted to know why or how bilirubin can cause itch. So first of all, I just tell you how do we study itch in mice, and it turned out it's very simple. We just inject whatever itchy chemicals into the back of the skin in the neck in the mice, and like a histamine or bilirubin, and you can watch them for record them for a half hour. As you can see here, the mice actually can elicit this characteristic scratching behavior using their hind paw and scratch at high frequency, about 10 hertz. So you can just videotape for 30 minutes and count how many times they scratch and quantify that. And so Jimmy actually did the experiment. So he inject bilirubin into the mouse skin at the increased dose. You can see here, bilirubin can induce a dose-dependent scratching behavior in wild-type mice, shown here. And he also pre-treated the mice with antihistamine. However, the antihistamine did not reduce bilirubin-induced itch, so suggesting that bilirubin caused non-histaminergic itch. And then he wants to know whether there is a bilirubin receptor exists in the mouse genome. So he actually screened all the mouse MRGs and human MRGs against bilirubin and using the standard calcium imaging as a readout for GPCRs. As you can see here, he found mouse MRG A1 and human genes called MRG X4 and show a pretty nice dose response curve in response to bilirubin stimulation. And consistently, and we also did a binding assay, and we found bilirubin can directly bind to these two receptors at the EC50 around the micromolar range. So this is the range you can find in the cholestatic patients. And it turned out most of the MRGs are low affinity. So the micromolar EC50 are low affinity and broadly tuned the receptor. So later on, 3D structure actually can show the why this receptor are low affinity and broadly tuned. So then Jimmy found this receptor is also specifically expressed in a subset of DRG neurons as shown as a green staining here. And then he also tried see whether bilirubin can really excite the neuron by using a patch clamp recording. As you can see here, bilirubin can induce a very robust action potential in the wild-type DRG neuron. However, if you apply bilirubin into the MRGA1 knockout DRG neuron, there's no activation at all. And consistently, if you just inject the bilirubin into the MRGA1 knockout mice, you see the bilirubin-induced scratching is significantly reduced. So suggesting the MRGA1 mediate bilirubin-induced itch. So those are the injecting the exogenous bilirubin. Here, Jimmy actually developed a cholestatic model for mice. So he actually used a chemical to damage the liver to block the excretion of bilirubin. So as you can see here, and after those liver damage or this cholestatic condition, there is a significant increase in the bilirubin level in the mice, in both wild type and MRG knockout mice. However, these mice actually can develop a spontaneous scratching because accumulation of bilirubin, presumably. So you can see the wild type mice, after the liver damage, they scratch significantly higher than the control mice. And however, the MRGA1 mice scratch much lower than the wild type mice. Again, this data suggests the MRGA1 actually mediate cholestatic itch. And then we collaborate with physicians. They actually collect the serum from several cholestatic patients. So this patient actually suffered a very severe itch, and their bilirubin level are significantly increased as we measured. And then Jimmy actually can inject this serum either from the healthy controls or from 6 cholestatic patient individuals and into the wild type and also MRGA1 knockout mice. And the healthy control serum did not induce significant scratching between the wild type and knockout mice. However, for the cholestatic patient serum, they induce a significantly higher scratching compared to the scratching in the MRGA1 knockout mice. So human cholestatic serum can cause itch in mice. So then we want to know what's in the serum really cause this itch, right? Is that bilirubin really the factor? So Jimmy actually using 2 different methods to deplete or block the bilirubin. First of all, he used iron chloride to deplete bilirubin from the serum. Another approach, he used anti-bilirubin antibody. In both cases, you can see at the green bar, the scratching are significantly reduced. So if you remove bilirubin from the serum, you can have a much less scratching, suggesting that bilirubin is a major contributor to in the serum to cause itch. So here, later on, Jimmy and several other labs showed the human version of this receptor not only binds to bilirubin, but it can also activate another lipid called the bile acid. So bile acid has shown to be a major factor for the cholestatic itch. Now we identify a novel bile acid receptor mediate bile acid induced cholestatic itch. So just summarize this part of the work. So we found the mouse MRGA1, human MRGX4, a bilirubin receptor, and human MRGX4 is a bile acid receptor, and they all mediate cholestatic itch. And now I want to switch the gear a little bit to talk about other MRGs. We found some other MRGs not expressed in sensory neuron, but rather expressed in the innate immune cells like mast cells, neutrophil, and the basophils. And so today I will tell you several stories about mast cell receptors and also neutrophil receptor. And they play an important role in pain and itch and also a host defense. So what is mast cells? So mast cells are the first responder cells upon infection and injury. So they are the tissue resident cells. You can find them in the skin, in the many other tissues. And so the major characteristic of this type of cells are they can, when they activate, they can degranulate and release the famous histamine to cause allergic reactions. And they also can release serotonin, protease, and cytokines, and to recruit other immune cells and promote inflammation. And so there's two major pathways to activate mast cell and degranulate mast cell. One major pathway is called the IgE and Fc receptor. So this is an antibody IgE, and you can have an antigen-specific IgE binds to the antigen and cross-link the Fc receptor to induce mast cell degranulation to cause classic allergic reactions. And then there's another major pathway people noticed for decades, and that is the pathway activated by this so-called positive charge substance, collectively called basic secretagogues. So basic means a positive charge, secretagogues means make mast cell to secrete. And there's many positive charge peptides and compounds can activate mast cell to degranulate. And however, the receptor is unknown for many years until our discovery. So a former postdoc, Benjamin McNeil, actually identified this basic secretagogues. And so he found this MRGB2 is exclusively expressed in mouse mast cells. And so we generate the MRGB2 Cre lines and use the Cre DT tomato to label the MRGB2 expressing cells. And we found 100% of this receptor are expressed in mast cells and shown using another marker called evidence. So there's 100% overlap. And you can find these positive mast cells in many tissue, including skins, airway, and heart. And this is also consistent with the online gene chip data. So people find by sequencing that mouse MRGB2 are exclusively expressed in mast cells and not in any other tissue, not in DRG either. And later on, people find the human homologs called the MRGX2 is also specifically expressed in human mast cells. So to make a long story short, and Ben actually used multiple approaches and find this basic secretagogues shown here can specifically activate these two receptors. For example, compound 48 is the most commonly used secretagogue, so degranulated mast cell. It can activate both receptors, and the substance P is a famous neuropeptide. It's also positive charged, can also activate these receptors. And besides these basic secretagogues, and Ben also found many other drugs can cause anaphylactic reactions, as shown here, can also activate these receptors, okay? And so these drugs are commonly used in the clinic to treat different kind of conditions. And so what is the anaphylactic shocks? So this is a relatively common conditions, especially during surgeries. So it can be life-threatening. So you can have a very sudden drop in the body temperature and lower blood pressure and short of breath, and can cause by food, venom, and medications. So it's pretty serious condition. And to our surprise, and all those drugs cause anaphylactic reactions can robustly activate wild-type mast cells, nearly 100%, all the mast cell response to those drugs. However, in the Mrgp2 knockout mast cell, they don't respond at all. So it's very clean result. And you can actually induce anaphylactic reaction by tail vein injection, one of the antibiotics called ceprofloxacin. So if you tail vein inject a high dose at very quick, shorter time, you can actually see the anaphylactic reactions. So here just show the body temperature is dramatically decreased within a few minutes in the wild-type mice. And many of these mice actually died because of shock. And then if you look at the knockout, they are fine. So none of them actually killed by these anaphylactic reactions. So this data indicate this mast cell specific receptor Mrgp2 mediate drug-induced anaphylactic reactions. So why this receptor can have so many ligands and activate them? So Brian Ross' lab at the UNC and another lab in China, actually they solved the 3D structure of this human receptor MRGX2 using cryo-EM. And they found this binding pocket, this receptor, has very shallow, so it can fit a lot of ligand into it. So the shallow binding pocket fit the idea of the promiscuous receptor. And they also find the negative charge residue actually can interact with the positive charged ligands. So basically, there's 3D structure consolidate with our biology data pretty well. So then we also want to know whether this mast cell receptor play a role in pain sensation. So former postdoc Dustin Green want to know whether this receptor, the mast cell receptor, have a role in pain or not. So as you know, substance P is a key neuropeptide mediate a process called neurogenic inflammation. So when you have a tissue injury, the injured nerve actually can secrete neuropeptides like substance P. And then substance P can activate nearby mast cells to induce inflammation by secreting cytokine, chemokines to recruit other immune cells. So this process is called the neurogenic inflammation because the inflammation is caused by nerve injuries through the substance P release. So the dogma in the field, in the pain field, suggests there's a canonical receptor or conventional receptor for substance P called the tachykinin receptor or NK1 receptor, function as a substance P receptor mediate neurogenic inflammation. However, the antagonist, the clinical trial for the antagonist NK1 failed with a very disappointing outcome because they don't have any analgesic effect. So what is going on here? So Dustin just performed a very simple experiment. He wants to see whether the NK1 receptor is really expressed in mast cell or not. As you can see here, the NK1, by using RT-PCR, he only detects NK1 receptor, the canonical receptor of substance P, only in the spinal cord, but not in the DRG sensory neuron, and more importantly, it's not in the mast cells. On the other hand, our receptor Mrgp2 are highly expressed in mast cells, not in DRG, not in the spinal cord. So it is MRGP2, but not NK1s expressed in mast cells. So consistently, when we apply substance P onto the mast cell, as you can see here, nearly 100% mast cell degranulate, whereas a knockout, MRGP2 knockout mast cell, there's no response at all to substance P. And then beside the degranulation, they also measure the chemokine release. So here, just show you a couple examples of CCL2, CCL3 released from mast cell degranulation, so induced by substance P treatment. So the wild-type mast cell release a lot of CCL2 and CCL3 upon substance P stimulation, whereas the MRGP2 knockout mast cell substance P failed to induce a CCL2, CCL3 release. And then they also, Dustin also inject the substance P into the skins and see the new immune cells recruitment. So here we use 3 different group of mice, wild-type mice, the NK1 knockout mice, and MRGP2 knockout mice. In both wild-type and NK1 knockout mice, substance P injection into the skin can induce a significant increase in immune cell recruitment like CD45 positive cells, neutrophil recruitment, and also monocyte recruitment in both knockout, NK1 knockout and the wild-type mice. However, in the MRGP2 knockout mice, substance P failed to induce immune cell recruitment. So the substance P promote immune cells recruitment through our receptor, but not from the NK1 receptor. And then besides injecting just substance P into the skin, Dustin also used this hind paw incision model, which I'm gonna show you the actual picture in the next slide. So basically, he just made a small cut into the hind paw and induce injury and the inflammation, and then using a flow cytometry to measure the immune cell recruitment from the tissues, from the hind paw. And we found wild-type mice, after injury, there's a significant increase of immune cell recruitment, whereas the MRGP2 knockout mice, the recruitment are significantly reduced. So just show you the actual picture here. So this is the incision model, wild-type and knockout and hind paw. And so here the picture illustrates this immune injury-induced inflammation is very prominent in the wild-type mice, but it's significantly reduced in the MRGP2 knockout mice. And consistently, the injury-induced pain sensation, here we measure the mechanical pain, also thermal pain has the same phenotype. The wild-type show very robust or very severe injury-induced pain, whereas the knockout, the pain is significantly reduced. So suggesting the MRGP2 contribute to inflammation and the pain. So here, just summarize this part of the work. And so upon tissue injury, the sensory nerve can release a lot of neuropeptide like a substance P and activate nearby mast cells through the receptor MRGP2 to release chemokine cytokine to induce immune cells recruitment and induce neurogenic inflammation and the pain. In a parallel study, the former student Jimmy actually also looked at the MRGP2 in mast cell into the non-histaminergic itch. So as you know, histamine can also cause itch during asthma or allergy. And so Jimmy did a side-by-side comparison between these 2 major pathways, mast cell degranulation pathways. And he found upon the allergic reaction pathways, the mast cell actually release a lot of way more histamine and the serotonin. On the other hand, if you activate this mast cell with MRGP2 pathways, they release less histamine, serotonin, but more tryptase. And they actually activate a different set of sensory nerve nearby, so to induce non-histaminergic itch. So now I want to switch gear to talk about another biology we just discovered. So we found a defensing receptor in neutrophil, which can prevent skin dysbiosis and the bacterial infection. And this work is done by a former postdoc, Xinzhong. And so what is defensing? So the defensing are the antimicrobial peptides, and first identified from the frog skin in 1987. So defensing is very important for us, actually. It can directly kill the invading bacteria and the fungi. And there are more than 50 defensing peptides and in human and mice. And it's expressed in the skins and also on the GI tract. And so they can directly kill the bacteria because they can form this helix and then insert into the negatively charged bacterial cell wall to make a hole to kill the bacteria. So people actually can design antibiotics based on the structure of defensins. And however, due to the gene redundancy, because there's 50 of them, and lack of genetic tools, the in vivo mechanism of defensing action is not unclear. So especially we don't know whether defensing, besides can directly kill the bacteria, can they actually also signaling our host cells? If that's the case, what's a host cell receptor for defensins? It's unknown. So how to really solve this problem? There's 50 defensing genes in the genome, but they all clustered on the mouse chromosome 8. So Xinzhong actually take effort to make a conditional knockout line by introduce a LOXP site into the each end of the defensing cluster. And so this process takes about 2 years, so it's a very long process. And it turned out really well, so it actually worked. So by introducing the LOXP site into the at the end of each end of the defensing gene cluster, and then she crossed with a keratinocyte-specific Cre called the K14 Cre, and then actually the deletion worked. So that's pretty amazing. So we can delete all 3 million base pair altogether, about 50 defensins altogether. Just showing the genomic PCR show that the whole cluster actually deleted. So you can have this PCR product made. And then she also looked at the mRNA for defensing. So here she used control wild type skins or the wild type skin infected with Staph aureus, the bacteria or SA. You can see after infection, there's a strong induction of a defensing expression. However, in the defensing cluster knockout mice, the defensing expression is completely gone. So here just show the immunostaining of the defensins. And so she used antibody to look at the defensins. So without infection, there's not much defensing expression. After the Staph aureus infection, you can see it turned on. And however, in the defensins knockout skins, there's not much defensing peptide expression at all. And to make a long story short, and Xinzhong also identified another MRG member called MrgA2 is a defensing receptor and specifically expressing a neutrophil. Here, she just used the RNA scope, the red signal, to look at the MrgA2 expression and use the Ly6G as a marker for neutrophils. And she found about 40% of neutrophil wild type mice show this the MrgA2 expression in neutrophil. However, in the MrgA2 knockout mice, the neutrophil expression is completely gone. Okay. Then she also did the neutrophil degranulation assay for this elastase release, and she found defensins can induce a dose response elastase release from neutrophil in the wild-type neutrophil. However, in the MRGA2 knockout neutrophil, the elastase release is completely eliminated. So suggesting the MRGA2 is a defensing receptor expressed in neutrophil. So a healthy skin can maintain a balanced and a diverse microbiome on their skins, which can prevent the bacterial infection. However, if you have any pathological disturbance, they will lead to outgrowth of pathogenic bacteria at the expense of other diverse commensals. So here, Xinzhong actually performed this 16S ribosomal RNA sequencing for the bacteria on the naive skins, comparing the wild type, the defensing knockout, and also the defensing receptor MRGA2 knockout skins. She found on the wild type skins, naive skins, the diversity of bacteria is very high, whereas in the knockout skins, the diversity of the microbiome is significantly reduced. And then look at the individual bacteria strains. She found in the wild-type mice skins, there's a very diverse and balanced the bacteria, different type of bacteria. However, in the knockout, there's an expansion of pathogenic bacteria. For example, this green one's Staph locutus is way expanded compared to the wild-type mice, suggesting without this receptor or defensins, and the mice has a dysbiosis, so the bacterial composition is totally changed. And beside the naive skin, and Xinzhong also infected the skin with by the exogenous Staph aureus. So she can either paint the bacteria onto the skin or inject the bacteria onto the skin, either wild type or the receptor knockout or the defensing knockout. And in the wild type skin, you can see the lesion size caused by bacteria and also the bacterial load in the wild type are significantly lower than the knockout animals many days after the infection. So here, just show you the examples, the representative images. So as you can see here, the knockout after the injection, the bacteria lesion size is a lot bigger than the wild-type mice, whereas using the bioluminescence imaging, we found after the infection, both knockout showed a significant higher bacterial load compared to the wild-type mice. So this data indicate without the defensing or without the defensing receptor, the skin cannot fight well with a bacterial infection, so you have a way severe infection afterward. So one of the hallmark of the Staph aureus infection is forming this neutrophil abscess. So here, Xinzhong did the skin histology 24-hour after the Staph aureus infection. So in the wild-type mice and knockout, she used Gram staining to look at the bacteria, shown here. And then for the Lysix-G to look at the neutrophil cells. And as you can see here, in the wild-type mice, after Staph aureus infection, the bacteria actually confined in a small region and surrounded by neutrophil. So actually, neutrophil actually attacked the bacteria to confine them into this abscess. However, in the both defensive knockout or the receptor knockout, there's not much neutrophil aggregation, and the bacteria actually spread in a large area. So that's why you have a larger lesion size shown here, a higher bacterial load. And here just show the quantification of neutrophil recruitment. And you can see here, wild-type mice has a lot of way more neutrophil recruitment where after the infection, whereas in the knockout, the neutrophil recruitment is significantly reduced. So both defensing and the receptor really mediate a neutrophil recruitment and the abscess formation. So mechanistically, we found after the bacterial skin infection, the neutrophil actually can release cytokines or chemokines. Here we just show you 3 different examples like IL-1 beta, CXCL1 or 2, and we found both in the knockout, in the wild type after infection. In all these chemokine and cytokine are significantly increased, released by the neutrophil, and then the knockout. And both knockout, these cytokine chemokine release are significantly reduced. And so they mediate the cytokine and the chemokine release. So here, just summarize this part of the work. So we think in the healthy skin, there's a low level of defensing, and they actually keep this diverse and balanced microbiome maintained on the skins and on the healthy skins. And there's also some skin resident neutrophil keep the healthy skin intact. However, after the bacterial infection, and this balance is disturbed, and then there's a upregulation of defensins increased, and then they actually activate the skin resident neutrophil and through the MrgA2 receptor, and they release interleukin-1 beta or CXCL1 or 2, and they can recruit more neutrophil infiltrate into the area to form this neutrophil abscess to confine the bacterial infection. So without the defensing and without the receptor, and this process is disturbed, and then you have a way worse infections. So here, I just want to show you what we're gonna do next. So as I mentioned, we found the mast cell receptor, specific receptor, and its function receptor for many neuropeptides like substance P, and play an important role in the neurogenic inflammation and pain and itch. And now we are working on the role of this receptor of mast cells in the ischemic stroke, and also in the fibromyalgia pain and the multiple sclerosis. So right now we are using mouse model for this disease to look at the mast cell and the mast cell receptor. And hopefully in the future, we can develop a small molecule antagonist blockers to treat many of these diseases. In fact, the phase 2 clinical trials is going on right now to testing some of these conditions using the MRGX2 antagonist. And for the A2 project, so again, this is a defensing receptor. Now we are moving into the brain to look at the brain infection like a meningitis and see whether defensing play a role there. And also we want to know whether the defensins and the MRGA2 play a role in the psoriasis. It's a condition where you can see a strong upregulation of a defensin in the psoriasis. So as mentioned, this receptor play a diverse role in many basic biological process and also disease, and from pain, itch, and innate immunity. And so we only deorphanize half of this gene family, and still many of them we don't have clue. We don't know where the expression and the ligand, so there's still a long way to go. And thank you very much for your attention. So here is the people who have done all the works. Thank you.
Oh, question?
Yeah.
Fantastic work.
Yeah, thank you.
2 related questions.
Yeah.
Sorry if I missed this. But given that X4 can respond to bilirubin as well as bile acids,
Yeah.
What is the evolutionary advantage to inducing a niche response to when there's like liver distress?
Yeah, so we don't know whether this receptor have any physiological roles, right? So recently we published a paper showing this also receptor for a phospho group. So we identified many phospho group drugs can activate this receptor. It turned out the adding phospho group is a common way to in drug development to induce water solubility. So people actually intentionally adding a phospho group into a difficult drugs to dissolve in water, but at the same time, it causes itch as a side effect. So, so we found those itch side effects actually caused by this receptor also. And, but from that, we actually found some other lipid, endogenous lipid, phospholipid, like geranyl diphosphate, can activate this receptor at a very low EC50, like a nanomolar range. So maybe that can give us some hint whether this receptor can sensing more endogenous phospholipid or something. So we are working on that. Yeah. Go ahead. You have second question or?
You sort of answered it. That are those the neurons that express X4, are they known to respond to any other stimulus? So it seems like the receptor itself.
Yeah.
Thank you.
Yeah. Go ahead.
Maybe at the same point,
Yeah.
from your data is though the bilirubin neurons that respond to bilirubin are
Yeah.
multimodal.
Yeah.
So I'm wondering if you looked at other ligands in terms of what else might act.
Yeah, so these neurons can activate bilirubin, bile acid, some other itchy substance. And in mice, it turned out there's 3 different itchy sensing neurons. And they called NP1, NP2, NP3. And so in mice, there's a small subset of itch neurons. However, in human, it's a little bit complicated. Maybe they are more converged into the one large subset, and one neuron can respond to multiple itchy substance. That's possible too. And somehow the mice has like more dedicated itch neuron now.
So it seems that there's those neurons, those primary sensory neurons,
Yeah.
might be mechanical sense.
Yeah.
Too.
Yeah, they do express piezos. So, but people actually in mice at least identified mechanical sensitive neuron too, mechanical itch neuron actually. Yeah, yeah. Go ahead.
So, I mean, first off, thank you for coming to speak with us.
Yeah.
I learned a lot. I'm kind of curious. I know at the end you touched on antagonists for Mrgprs for fibromyalgia.
Yeah.
So,
B2, sorry, B2.
yeah. So I'm kind of curious. Do you think there's like a hyperexpression going on in those patients or what?
Yeah. So this receptor we call this called Mrgx2, the B2, sorry, for the mast cells. And as I show you at the RT-PCR, the receptor is already expressed very high level in those mast cells. And we don't think the expression in particular cell gonna go up and more because they're already very high expressing genes. So maybe the number of new mast cell can increase or they aggregate. That's possible. Yeah, people actually has the data showing the fibromyalgia. We have our collaborator have unpublished data showing the fibromyalgia patient skins actually has a lot of mast cell aggregates. So yeah, so there's many companies working on this receptor antagonist right now. Yeah.
Thanks for the great talk. I was curious about the defensive.
Yeah.
Hear what cell type they're coming from. I think they're signaling to the neutrophil. Is that right?
Yeah. So basically, we showed there's 2 actions. Other people show there's a direct killing action to the bacteria, but now we show they can activate the neutral field, and they made mainly from the keratinocyte. So that's a beta defensins. There's also alpha defensins. Alpha defensins is more in the GI tract. They are made of different type of cells. And so we found actually the meninge cells also make defensins. So that's why we are looking at the meningitis right now. Yeah. Any other questions? Yeah, thank you.